Okay, so 18.4, individual bacteria respond to environmental change by regulating their gene expression. So this is about how bacteria turn on and off genes. So a bacterium can tune its metabolism to the changing environment and food sources. And it can do this by one of two different ways. There's two levels. They can adjust the activity of metabolic enzymes, and they can regulate the genes. So they can decide whether or not the enzymes are going to work or not, or they can turn on and off genes that even make the enzymes in the first place. And so that's what we're looking at here. So this is figure 1820 in your book. It's titled Regulation of a Metabolic Pathway. Okay, so there are two sides to look at this. Regulation of enzyme activity, regulation of enzyme production. So let's write something off to the side here. So bacteria, like E. coli in our intestines, need something called tryptophan to survive. So if it's in the environment, they don't need to make tryptophan. But if it's not, then they would need to make it or else they die. And so this pathway that we're looking at here is a pathway where the bacteria starts out with a precursor molecule and goes through a series of reactions, ends up with tryptophan as the end product. So you need to make tryptophan only when you don't have it. And so that pathway, though, is catalyzed by 5 different enzymes, and each one of those enzymes are made by 5 different genes. And so if we look at the regulation of gene expression here, notice here at the end, tryptophan, if you have an abundance of tryptophan, notice this negative here. It's going to stop the transcription and translation of these genes. So let's write here that the abundance excuse me, the abundance of what? Of tryptophan can turn genes off. All right, because that makes sense, because if you have enough tryptophan, then you don't want to go through this pathway to make more tryptophan, so we want to turn that off. So one way that we can do that is turn the genes off so we don't make the enzymes, and if you don't have the enzymes, you can't make tryptophan. All right, so that's the first thing. So that's the regulation of enzyme production. On this side here is the regulation of enzyme activity. So in this case here, an abundance of tryptophan, the tryptophan acts as an inhibitor, all right, of the first enzyme. So that's why it was over here of enzyme 1. So what happens is if you have an abundance of tryptophan, let's write abundance here, tryptophan, that tryptophan will actually act as an inhibitor to this enzyme. And if this enzyme doesn't work, guess what? The rest of the reaction is not going to happen. So it kind of turns itself off. All right. So that's feedback inhibition, is stopping that pathway from happening. Okay. All right. So let's turn our page. So bacteria, in turning genes on and off, have these things called operons. This is unique to bacteria. In bacteria, genes are often clustered into operons composed of, it has an operator, which is an on-off switch, a promoter region, and the genes for making your enzymes. So an operator or operon can be switched off by a protein called a repressor. A co-repressor is a small molecule that cooperates with a repressor to switch an operon off. So let's look at this particular picture. So this picture right here, I'm going to write here as figure, if you want to refer back into your textbook, is 1821, and it's called the trip operon. Trip stands for tryptophan, which, remember, the bacteria needs to survive. All right, so let's look at what we're looking at here. Okay, so this is the trip operon right here. So we just saw it's made out of an operator, a promoter, and genes for making enzymes. So here you have, here's your operator region. It's part of your promoter region. So this whole thing is the promoter region, which is, remember, where our transcription factors and RNA polymerase binds. Remember we talked about that in chapter 17. And so RNA polymerase binds, and so this is the operator region. And here are your genes, and there are 1, 2, 3, 4, 5 genes. All right, kind of goes with over here. 1, 2, 3, 4, 5 genes here, coding for different enzymes. And so notice here when RNA polymerase binds, all right, it can go along and transcribe in the DNA. So this is an RNA polymerase that has already gone through there. And so notice here that it goes through and makes one mRNA. This is your mRNA strand from the DNA. One mRNA made in the 5 to 3' direction for all 5 enzymes. This is different than in eukaryotic cells like our cells. In our cells, we just do one gene at a time. And so these guys are transcribed all together. And so notice here, here's the start codon and a stop codon to make. And so when it goes to the ribosome to do translation, this represents translation, mRNA to polypeptide. This right here from the DNA to the mRNA is transcription. There's supposed to be an I in there. Transcription. All right. So when it goes to the ribosome, there's a start codon and there's a stop codon. So when the ribosome moves along here, and then you make one polypeptide, all right, one enzyme. Then right after that's another start codon and a stop codon. So it starts and stops and makes a new polypeptide. Start, stop, and makes another one. So let's write that here. It doesn't show you here. There would be another start here. And then this would be another stop, and then there would be another start. It's kind of hard to write in there. It's kind of small. But you get the idea. And so that's why you don't get these polypeptides, all one chain of polypeptides. You get 5 different polypeptides made from one mRNA because of the start and stop codons. And so it stops making that polypeptide, and you get your finished one before it starts making the other one, and so on. And so you get all the polypeptides that are useful for tryptophan synthesis. So why is it this beneficial for the bacteria to make all 5 at once? Because all 5 of these enzymes are used in the same pathway. So you don't really ever are going to need enzyme 1 by itself or enzyme 2 and so on. In fact, that's useless unless you have all 5. This pathway can't work. So therefore, why not make all 5 at once? So why not have them grouped together in clusters as far as you doing the same function, and then you make all 5 enzymes at once, and then you can do the pathway and make tryptophan. But remember, you only want to make tryptophan if it is it's needed and is not in the environment. Otherwise, it would be wasteful. So then let's look at what this part of the picture is looking at. So now this is showing you, this right here means that in the DNA, but far away from this operon here that's making these enzymes for tryptophan synthesis. So over here, you have another gene. The R stands for regulatory gene. So it has its own promoter and it's another gene. So when this goes through transcription and translation, mRNA to protein, it makes an inactive repressor. And so a repressor molecule, we just said, an operon can be switched off by a protein called a repressor. And so how this works is, if you notice this part, let me get a different color here, this part of the repressor, this is the part that's important. It's the shape. That's what makes it inactive, because this gene makes a repressor that's going to regulate or turn on and off the operon. So this coincides with the operator. Do you notice here the operator has a specific shape? Right now, this repressor cannot fit into the operator. Hence, it's inactive. So it's inactive, so it's not able to be successful. It's not repressing these genes, so these genes are turned on. So let's write here that this regulatory gene makes the repressor, codes for making the repressor. And it's inactive. Alrighty, so let's look specifically about what makes it active. That's what this next picture shows. Alright, so this is still the regulatory gene right here. This is your trip operon. Right, this whole thing. This right here is your promoter region right here, and this is that operator that I was just talking about. All right, so that's this part right here. So notice here your regulatory gene makes the repressor in the inactive form. So it cannot bind, so that's what I was talking about here, it cannot bind with the operator. But look at what happens is let's think about if there's tryptophan present. So if there's a lot of tryptophan, the tryptophan can actually bind here. All right, so this right here, the trip binds to the repressor. And look at what happens. It binds there and boom. What happens to the shape? It changes the shape and it makes it into the active repressor. So that's right here, to make it active. So from here to here, the tryptophan binds, and now look at this has changed shape right here. It's now active, and what it can bind right into that operator region. And now look at what happens is that now when RNA polymerase, if you have this operator region with the repressor bound there, RNA polymerase can't get back to it. It acts as a physical blocker and no RNA is made. And so that so therefore these genes to make tryptophan are turned off. And why? Does that make sense? Tryptophan changed the shape because if you have tryptophan, if there's enough that it can change the shape of this and bind to it, that means there's enough tryptophan that the bacteria doesn't need to make its own tryptophan. It's already present. So therefore, we want to turn those genes off because tryptophan is already present. So trip is already present. So it would be wasteful so it will be wasteful to make tryptophan. Wasting energy, that's what you're wasting there. So it turns that gene off. So let's turn the page here. So these operons can be divided into categories. A repressible operon is one that's usually on. Binding of a repressor shuts repressor to the operator shuts off transcription. All right. Is that true about the trip operon? Is it usually on? The repressor is made in the inactive form. It can't bind, so it's by default on, making the enzymes to digest or to make tryptophan. When you get tryptophan in the environment or in the bacteria, it turns the repressor in the right shape and the genes are turned off. So it's normally on. So the trip operon is what we call a repressible operon, meaning that it's on and it's repressible. It has the ability to be repressed or to stop or to be turned off by the presence of what you're trying to make in the first place of tryptophan. All right, so it's a repressible operon. Now, we're going to talk about another type of operon. An inducible operon is one that's usually it's off. A molecule called an inducer inactivates the repressor and turns on transcription. So a classic example of an inducible operon is called the lac operon, which contains genes coding for enzymes in hydrolysis and metabolism of lactose. Hydrolysis, breakdown. So the last one we were talking about making tryptophan, and this one we're talking about breaking down lactose. Lactose can be a food source for bacteria. It is a disaccharide. All right, so looking at the lac operon here. So here is the regulatory gene. Here's the promoter region. Here's the operator, and here's your genes. And so normally, here's your regulatory gene makes the repressor. The repressor is made in the active form. So when the regulatory gene makes the repressor, that repressor can automatically go, it's already made in the right shape, that it can automatically go and bind to the operator region. Like this, it has the right shape, and so therefore RNA polymerase can't bind, and therefore the gene is usually off. So I'm going to write here that it's usually off. Why? Because the repressor is made in the active form. Okay, and this is, let's write here that this is different than the trip operon, which was inactive in the inactive form, an inactive repressor, unless trip was present. So this is active, so it's normally turned usually off. All right, off. So let's look at the picture below. This shows you just the rest of the lac operon here. So then, so this shows you lactose absent. So when there's no lactose, this is turned off. Why? Because if you don't have lactose, then you don't need to make the enzymes to digest it. Because remember, we're making enzymes, which contains the genes for coding for enzymes to break down lactose. And so if there's no lactose there, it would be wasteful to make the enzymes to digest it. So therefore, let's look at when lactose is present. And so here's your regulatory gene. Here's the lac operon. And here, this only shows you part of it. This shows you the rest of the genes. So here's your regulatory gene, makes the mRNA. You make the repressor in the active form. All right. But if lactose is present, then it binds to the repressor and makes it in the inactive form. So let's write a couple of things. Notice it says allolactose. All right, so let's write what that is. This is, let's put over here, an isomer lactose formed in small amounts. Formed in small amounts when lactose is present. Okay. So if lactose is present, there's going to be this allolactose present, and that can bind to it and inactivate the repressor. All right, so it changes shape here. So an inducer, let's write here that inactivates the repressor by changing its shape. All right, so it's inactive, which means now that it can't bind. So now it can't bind to what? What does it bind to? What is this thing up here? the operator. And if it can't bind to the operator, the genes are on. So therefore, there's nothing physically blocking. Like here, when it was made and it was not making the enzymes, now there's nothing physically blocking because the allolactose is binding to it. So now RNA polymerase can go through, make the mRNA. It works the same as the previous, where you get each gene has a start and a stop codon, and you make 3 different enzymes. So all 3 of these are enzymes to digest lactose. And that makes sense that now you're making these enzymes and that this operon is turned on. Why? Because lactose is present. All right, and so therefore, because lactose is present, it makes sense to make the enzymes to digest it, and so therefore this gene is turned on. Okay. So let's flip it over. So a little bit different there than the trip operon. So this is an inducible operon. Why? Because it can be induced or made to turn on or made to produce something. So it's normally off and can be inducible only if lactose is present. So inducible enzymes like this usually make enzymes to digest or break down stuff. What kind of pathway is that? Inducible enzymes usually function in, is it catabolic or anabolic? It is catabolic. Catabolic is breaking stuff down. So inducible enzymes are usually involved in catabolic pathways. Repressible means that they're able to be repressed, which means they're normally on. Repressible enzymes are usually function in anabolic pathways where you're making something. So in this case here, they were making tryptophan. All right, making tryptophan. So regulation of the trip and lac operons involves what we call negative control of genes because the operons are switched off by the active form of a repressor. So the repressor turns it off. Okay. There's also, last but not least, positive gene regulation. So we're going to build on what we know about the lac operon that we just talked about. So some operons are also subject to positive control through a stimulatory activator protein, such as, and we call it, it's catabolite activator protein, or they call it the CAP protein. When glucose, which is a preferred source of E. coli, is scarce, the lac operon is activated by the binding of CAP. So what this means is if you have a bacteria and they're given glucose and lactose as 2 choices to quote unquote eat or use for energy, they prefer glucose if they're given the choice. Now, if there's only lactose, they'll use lactose, but you get more energy from glucose. Glucose is a monosaccharide. You don't have to break it down. They can use it right away for cellular respiration or for energy. Lactose, on the other hand, is a disaccharide, and you have to, so 2 sugars hooked together, you have to break it down first before you can use it for energy. And so therefore, glucose is the preferred source if given a choice. So therefore, the bacteria have an option as far as which one they're going to use for energy. And they have to have a system in place to say, okay, if glucose is present, we're going to use glucose and not lactose, and vice versa. All right, so when glucose levels increase, so in the environment, glucose levels increase, so there's more and more glucose available for the bacteria, this CAP protein detaches from the lac operon and turns it off. So this picture is going to help to explain that process. So we're adding on, like I said, to what we know already about the lac operon. So this is figure 1823 in our textbook. This is positive control of the lac operon by CAP. All right, so let's look at this. This is your regulatory gene. And this whole thing right here is your lac operon. And this is your promoter region with the operator and the spot where RNA polymerase binds. So that's what we're looking at. So let's look at our scenario down here. When it says letter A, lactose is present, glucose is scarce. So let's go back here. Remember, if given a choice, they'll always choose glucose first. But saying that glucose is scarce, there's not a lot of glucose, but there is lactose present. So is it going to want to digest the lactose? Absolutely, because that's going to be its only energy source, is to digest the lactose. So therefore, what do you need to digest lactose? You need enzymes. And so therefore, you want this operon to be turned on, because remember, the operon makes enzymes to digest lactose. And if lactose is present, you definitely would like that on. All right, so, and then it says glucose is scarce. And then it says cyclic AMP level high. So let's talk about that. When we break down ATP, we break it down into ADP, right? Plus an inorganic phosphate. But if you don't have very much glucose, then what the cell is going to do is take that ADP, and instead of, if there's no glucose, then instead of putting the phosphate back onto ADP to make more ATP, that's not going to occur because you need energy, glucose, to do that. So if glucose is scarce, you're going to break down ATP, you're not going to remake it, and you could possibly then take ADP and break it down even further to AMP. And so cyclic AMP is a version of that. So when you have this cyclic AMP present, what that means is, wow, we really need some energy because we're even using the ADP now for energy. So let's look at this up here now. Here's your cyclic AMP. And that's right here. Let's say that this accumulates. We just talked about this, but let's write it here. Accumulates when glucose is scarce. Oops, scarce. There we go. Accumulates when glucose is scarce. So glucose is scarce, lactose is present. So we need some energy, so we need to digest this lactose. So we need these genes to be turned on. So what does cyclic AMP do? Notice here, it binds to, it says inactive cap. So inactive cap, it has this shape here, but notice here that cyclic AMP can bind. So cyclic AMP binds to cap and activates it. It's now in the active form. Why? It has a specific shape. Change the shape, made it active. Notice what it does. It binds to the promoter region and then actually allows RNA polymerase to bind, and therefore RNA polymerase can transcribe and go through transcription there. So let's put this active cap. What it actually is, it's an activator of transcription. The active cap. So it activates, it binds, that's the cap binding site, and RNA polymerase can bind. And notice that there's nothing bound to the operator region. Why? Because lactose is present. So remember from the previous picture, when lactose is present, there's that allolactose present. Allolactose binds to the repressor, keeps it in the inactive form, so the shape cannot bind, and therefore it can't stop transcription. So therefore, when lactose is present and glucose is scarce, what do we want to do? We want to turn those lactose genes on. So two things trigger that to turn on. Cyclic AMP activates the cap protein, so they can act as an activator for transcription. RNA polymerase can bind. It's not blocked because lactose is present, and therefore the repressor is inactive. So these genes will be turned on. All right, so the mRNA is synthesized. So then let's look at what happens with the next one, lactose present and glucose present. So now you have, here's your bacteria, you have glucose and lactose present. Remember, glucose is the preferred food source. So how does it know to do glucose and not lactose? Well, if glucose is present, guess what? There's going to be no cyclic AMP. So no cyclic AMP, which means there's going to be nothing to bind to the cap protein right there. So the cap doesn't change shape. It can't bind to the promoter region, which means that RNA polymerase can't bind. So even if there's lactose present and it can't bind to the promoter because it's making the repressor inactive, just like before, it's not binding to it. These genes are not turned off because not only do we need lactose present, but we also want glucose to be absent in order to turn those genes on. So that's how the bacteria chooses between glucose and lactose, is that even if lactose is present and it doesn't block that operator, because glucose is present, there's going to be no cyclic AMP. The activator can't bind, cap can't bind, and neither can RNA polymerase, and the genes, this little mRNA is synthesized, so the genes would be turned off, and that's how it quote-unquote chooses that. Okay, and that is the end of chapter 18.